Okay, hello everyone. I can see that everyone is very tired today. It's already 4:45, but I hope you can still bear with me for another 15 more minutes, or maybe less, depending on how fast I talk. So, here goes. In Peninsula Malaysia, the largest intertidal seagrass meadow can be found in Johor. And there is quite a lot of research has been done in Johor. But have you ever wondered where is the second largest intertidal seagrass meadow in Peninsula Malaysia? So, this is, let me introduce you to the Bank Seagrass here. Okay, so this Bank Seagrass is located at the Penang Island. It is the second largest intertidal seagrass meadow here in Peninsula Malaysia. So if you're wondering where Penang Island is, so let's look at the map here. So that is Peninsula Malaysia, and this is where the Penang Island is. And the seagrass meadow is located at the east coast of the Penang Island here. So there are three species that can be found here, which is first the Inhalus acroides, second Thalassia hemprichi, and the third one is the Halophila ovalis. So there are not many research has been done here, and there are lots of knowledge gaps. So this is why I'm standing here today to show you the importance of the second largest intertidal seagrass meadow to this island, yes. So this is my topic for today, unfolding the gem of Penang Island, that is the Bank Seagrass meadow, and its relation to water clarity. And by the way, my name is Asilah. I'm from University Malaya. Okay, this is my first time presenting in ISBW, by the way, so sorry if I'm being so very nervous right now. Okay, so what is the relationship between seagrass and water clarity? So we all know that seagrass is also as an ecosystem engineer. In fact, ecosystem engineer is like the middle name for the seagrass. So how does it relate to water clarity? So first, let's look at the seagrass helps to reduce the water current, which is by creating a drag when the water passes through it. And the leaf blade helps to trap the sediments. Eventually, it leads to the sedimentation and improve water clarity. So for my research, we will focus on the water clarity. But before that, this is an example at Rottnest Island, where you can see here the water is very clear at a very dense meadow, the eelgrass meadow here. So this is what we are thinking, that the theory that the seagrass actually helps in water clarity. So there are three key water clarity parameters here. First is the sedimentation rate, the light intensity, and water transparency. So these are the main key questions. First, does Bank seagrass improve in water clarity? And second, what are the elements of the meadow structure that plays a significant role in water clarity? And third, what are the considerations in measuring water clarity parameters in intertidal meadow? So in order to achieve the objective, let's look at the methods. The response variables, the water clarity parameters, which is the sedimentation rate, light intensity, and the water transparency. As for the exploratory variables, these are the meadow structures. That is the biomass, the shoot density, and the leaf area index. So we look at it one by one, how the samples were conducted for the response variables. As you can see here, this is the map. How to see it? Okay, so this is the Bank seagrass meadow. We only focus at the north of the meadow here, and we categorize it into three different zones. That is the outside zone, the edge, and the inside of the meadow. And to get the sedimentation rate, okay, here goes. Yeah, we use a sediment trap, not the conventional trap, that the erect type, but the J-shaped tube trap that is recommended by the Shield et al. Which the trap used for the intertidal to get the sedimentation rates for the intertidal zone. So as for the light intensity, we use a Hubble-pendant temperature light data logger that is being strapped on the sediment trap. So this is how the sediment trap looks like, and these are the triplicates. As for the water transparency, just a normal Secchi disc will be used for this sampling. So for your information, the location of the traps are randomly randomly chosen around the the zones, the outsides, the middle, in the edge and the insides. So they are just randomly chosen. As for the meadow structures. We use the seagrass corer, so this is the seagrass corer, which is used to sample the seagrass, and the samples were collected at the vicinity of where the sediment traps are located. So 3 samples for every species that were found there, and this, the biomass, leaf area index, and the shoot densities for all the samples were measured in that. So now let's focus on the findings. We go through one by one the sedimentation rate, the light intensity, and the water transparency. So for the sedimentation rate, as you can see here clearly that the sedimentation rate in seagrass zone is significantly higher compared to the area where there is no seagrass. Okay, so which supports the theory that the seagrass actually helps to improve the water clarity by increasing the sedimentation rates. So it shows here. And as for the light intensity, okay, initially we, like I mentioned, that we want to compare between the zones and also the area with seagrass and no seagrass. But unfortunately, we lost some of the loggers that's located outside of the seagrass, so I cannot really compare because there's not enough data. So that's why I have to look at between the edge and the inside of the seagrass. So these are the light intensity, and there's also significantly higher for the area at the edge compared to the inside. This is based on the light logger. Okay. But by the way, I forgot to mention that I only present the significant data here because there's quite more things to talk about, right? So, okay. Like I mentioned before, we want to see the relationship between the water clarity and the meadow structure. So the meadow structure, there are 3 variables I mentioned just now. So we do an analysis between the light intensity and the meadow structure, and the most significant one that can be found is the shoot density, which means that the shoot density is influenced more compared to the biomass and the leaf area index. That's like 74% of the light intensity variation can be explained by the shoot density. But the question is which seagrass, which species, because this area got 3 different seagrass, but we only focus on the extreme one, which is the smallest seagrass and the biggest seagrass, the inhalers and the Halophila ovalis. So we compared between the shoot density in Halophila ovalis and the inhalers or corridors. So as you can see here, there are obviously the Halophila ovalis influence the light intensity, not the inhalers or corridors here. So what are the reasons why are these small seagrass that actually helps in terms of light intensity? We said that light intensity is actually like water clarity. People thought that bigger seagrass actually helps to trap the sediments, but how come this small seagrass actually helps, not the inhalers one? So as you know, the intertidal zone, there's a lot of resuspension things occur, right? So this, in an area where there's a lot of Halophila ovalis, which means that the area are very dense and the rhizomes are extensive. There's an extensive rhizome network. So combined with the leaves and the rhizome, the Halophila ovalis is like creating a blanket on top of the sediments, which holds the sediments from reducing it from being resuspended. So that is why we thought that possibly that the area where there is a lot of Halophila ovalis have less resuspension occurs, and that is why the light intensity is quite high in the area where there is Halophila ovalis compared to the area with no Halophila ovalis. So I just recap, because of the extensive rhizome and because of the denser Halophila ovalis, it helps to hold the sediment and reduce the resuspension. As for the water transparency, you can clearly see here there's a significant difference between outside, edge, and inside. So this shows that the clarity actually improves as water passes through the meadow. It's real obvious here. So these are the conclusions. The middle bank seagrass improved water clarity from outside meadow towards inside meadow to almost 90%. This is based on the water transparency results. Okay. And the Halophila ovalis shoot density significantly influenced light intensity compared to the inhalers. And third, the water transparency is considered as a suitable parameters for water clarity in intertidal meadows, specifically the middle bank seagrass. Thank you for your attention.
Yeah, and thank you for your quick talk. I think it was informative. Are there any questions?
Thank you, that was an interesting talk. I was wondering why the water clarity results seem to be different to the light intensity results. If I understood the graphs correctly, the light intensity was higher on the edge of the meadow compared to the inside of the meadow, but the water clarity was better on the inside of the meadow than the outside of the meadow.
Oh, okay, wait. Let me just check back the data. Okay, the light intensity at the edge of the meadow is higher compared to the inside of the meadow. I will say here maybe there's another factors, not just the Halophila or any other species there, because at the edge of the meadow there is a lot of inhalers. There's a possibility that the area where we put the light intensity actually causes shading to the light intensity. So most probably that's because of that, because we can find lots of Halophila orbilis at the inside of the meadow.
And was there any differences in the depth between the two sites?
You mean between the edge and the inside? Not really. It's like almost flat.
Thank you.
All right.
Hi, thank you. Did you see any differences between the species on the sedimentation rates?
The what, sorry?
Any difference between the species? So Alophila and bare sediment, they had different sedimentation rates. What about the Halus? Do you take different species? You had 3 species, right?
I'm sorry, I didn't get you. What do you mean by space?
You had 3 species.
Uh-huh.
And
you mean the zone, the outside, inside, and the edge? Is that you mean?
yeah.
Okay.
Inside the meadow, you had 3 species of seagrass.
3 species? All right.
Yeah.
Sorry, okay.
Yeah.
Now I understand. Okay.
Did they have different sedimentation rates?
Sedimentation rate between, it's really hard for me to differentiate between the sedimentation rate between the species, because the mixed meadow, there are not really patches differently, so I put the trap not in depending on the species, but just randomly chose. So I cannot really compare between the species of the sedimentation rates.
Okay, thank you.
Any more questions? Oh no, that's fortunate because it's exactly the right time to move on to the next talk. So thank you again for your presentation.
All right, thanks.